Assalamu alaikum. My name is Umme Habiba. I am from Chemistry Department and topic of my presentation is polysaccharides and some other important carbohydrates. First of all, we see contents of presentation which include introduction, examples, synthesis of polysaccharides, functions of polysaccharides, and some important carbohydrates. So, what are polysaccharides? Polysaccharides are complex carbohydrates in which tens, hundreds, or even thousands of simple sugars are linked together through glycoside bonds. Because they have only the one free anomeric hydroxyl group at the end of a long chain, polysaccharides are not reducing sugars and don't show noticeable mutation. So, cellulose and starch are the two most widely occurring polysaccharides. In examples, first of all, we see cellulose. Cellulose consists of several thousand D-glucose units linked by 1 to 4 beta-glycoside bonds like those in cellulose. Different cellulose molecules then interact to form a large aggregate structure held together by hydrogen bonds. This is structure of the cellulose. Nature uses cellulose primarily as a structural material to impart strength and rigidity to plants. Leaves, grasses, and cotton, for instance, are primarily cellulose. Cellulose also serves as raw material for manufacture of cellulose acetate, known commercially as acetate trim, and acetate cellulose nitrate, known as gun cotton, which is the major ingredient in smokeless powder, the explosive propellant used in artillery shells and in ammunition for firearms. So the second is starch and glycogen. Potatoes, corn, and cereal grains contain large amount of the starch and a polymer of glucose in which the monosaccharide units are linked by 1 to 4 alpha glycoside bonds like those in maltose. So starch can be separated into two fractions, amylose and amylopectin, which are two isomers of this. And first we discuss amylose. Amylose accounts for about 20% of weight of starch and consists of several hundred glucose molecules linked together by 1 to 4 alpha glycoside bonds. This is structure of amylose, which is alpha 1 to 4 D-glucopyranoside polymer of the starch. Then we discuss about amylopectin. Amylopectin accounts for the remaining 80% of starch and is more complex in the structure than amylose. Unlike cellulose and amylose, which are linear polymers, amylopectin consists 1 to 6 alpha glycoside branches approximately every 25 glucose units, and this is structure of amylopectin, which is one of the isomers of the starch. Starch is digested in the mouth and stomach by alpha glycosidases, which catalyze the hydrolysis of glycoside bonds and release individual molecules of the glucose. Like most enzymes, alpha glycosidases are highly selective in their action. They hydrolyze only alpha glycoside links in starch and leave the beta glycoside links in cellulose untouched. Thus, humans can digest potatoes and grains, but not grass and leaves. And now we discuss about glycogen. Glycogen is a polysaccharide that serves the same energy storage function in animals that starch serves in plants. Dietary carbohydrates not needed for immediate energy are converted by the body into glycogen for long-term storage. Like the amylopectin found in starch, glycogen contains a complex branching structure with both 1 to 4 and 1 to 6 links. Glycogen molecules are larger than those of amylopectin, up to 1 like glucose units, and contain even more branches. Then we discuss about synthesis of the polysaccharides. What are the method of their synthesis? With numerous hydroxyl groups of similar reactivity, polysaccharides are so structurally complex that their laboratory synthesis has been a particularly difficult problem. Several methods have recently been devised, however, they have great simplified the problem. Among these approaches is the glycerol assembly method. This method, easily prepared from the appropriate monosaccharide, a glycerin is an unsaturated sugar with a carbon 1 carbon 2 double bond. To ready it for use in polysaccharide synthesis, the glycerin is first protected at the primary hydroxyl group by the formation of a slide ether and its two adjacent secondary hydroxyl group by the formation of a cyclic carbonate ester. Then the protected glycol is epoxidized. This whole process is expressed in this reaction. A glycine is converted to protected glycine, then an epoxide is formed. Treatment of the protected glycol epoxide is the present in the presence of zinc chloride as a Lewis acid with the second glycol having a free hydroxyl group causes acid catalyzed opening of the epoxide ring by SN2 backside attack and yields a disaccharide. The disaccharide is itself a glycol, so it can be epoxidized and coupled again to yield a trisaccharides and so on. Using the appropriate sugars to each step, a great variety of polysaccharides can be prepared. After these sugars are linked, the silyl ethers and cyclic carbonate protecting groups are removed by the hydrolysis. Then, among the numerous complex polysaccharides that have been synthesized in laboratory in Lewis, Y-hexasaccharide, a tumor marker that is tested as a potential cancer vaccine, and the structure of that is given in this figure. Lewis Y hexasaccharide. Then functions of the polysaccharides. Depending on their structure, polysaccharides can have a wide variety of the functions in nature. Some polysaccharides are used for storing energy, some for sending cellular messages, and others for providing support to cells and tissues. So storage of energy, many polysaccharides are used to store energy in organisms while the enzymes that produce energy only work on the monosaccharides stored in the polysaccharide. Polysaccharides typically fold together and can contain many monosaccharides in a dense area. Further, as the side chains of the monosaccharides form as many hydrogen bonds as possible with themselves, water can't enter the molecules, making them hydrophobic. This property allows the molecules to stay together and not dissolve into the cytosol. So it is one of the main advantages of the polysaccharides in the plants. This lowers the sugar concentration in a cell, and more sugar can then be taken in. Not only do polysaccharides store the energy, but they allow for changes in concentration gradient, which can influence cellular uptake of nutrients and water. Then cellular communication. Many polysaccharides become glycoconjugates when they become covalently bonded to proteins or lipids. Glycolipids and glycoproteins can be used to send signals between and within cells. Proteins headed for a specific organelle may be tagged by certain polysaccharides that help the cell move into a specific organelle. The polysaccharides can be identified by special proteins which then help bind the protein, vesicle, or other substance to a microtubule. The system of microtubules and associated proteins within cells can take any substance to its destined location once tagged by specific polysaccharides. Further, multicellular organisms have immune systems driven by the recognition of glycoproteins on the surface of the cells. The cells of the single organisms will produce specific polysaccharides to adorn its cells with. When the immune system recognizes other polysaccharides and different glycoproteins, it is set into action and destroys the invading cells. Then we see cellular support. Polysaccharides give cellular support. By far, one of the largest roles of polysaccharides is that of support. All plants on Earth are supported in part by polysaccharide cellulose. Other organisms like insects and fungi use chitin to support the extracellular matrix around their cells. Between chitin and cellulose, both polysaccharides made of glucose monosaccharide, hundreds of billions of tons, are created by living organisms every year. A polysaccharide can be mixed with any number of other components to create tissues that are more rigid, less rigid, or even materials with special properties. Everything from the wood in trees to the shells of sea creatures is produced by some of the polysaccharides. Simply by rearranging the structure, polysaccharides can go from storing molecules to much stronger fibrous molecules. So these are important functions of the polysaccharides. So we see now some other important carbohydrates except monosaccharides, disaccharides, and polysaccharides. In addition to the common carbohydrates, there are a variety of important carbohydrate derived materials. Their structural resemblance to sugars is clear, but they are not simple aldoses or ketosis. Deoxysugars have an oxygen atom missing, that is, an hydroxyl group is replaced by an hydrogen. The most common deoxysugar is 2-deoxyribose, a monosaccharide found in DNA. So 2-deoxyribose exists in water solution as complex equilibrium mixture of both furanose and pyranose forms of the sugars. So amino sugars such as D-glucosamine have an hydroxyl group replaced by an amine. The N-acetyl amide derived from D-glucosamine is monosaccharide unit from which chitin, the hard crust that protects insects and shellfish, is made. Still, other amino groups are found in antibiotics such as streptomycin and gentamicin. So these are some other important carbohydrates, except main classes of the carbohydrates, which have also um, important functions in our daily lives. Thank you.